Welcome back to the deep dive. I was reading through the source material for today, Murray's Medical Microbiology, Chapter 8, and I had this sudden, slightly terrifying realization.
Oh, yeah.
We spend our whole lives sort of vaguely worried about germs, right?
Yeah.
We carry hand sanitizer, we wash our food, we avoid that guy sneezing on the subway.
Which, to be fair, is generally good advice. You should probably still do that.
It is, absolutely. But it creates this illusion that we are these fragile, defenseless little things just constantly on the verge of being overrun. But when you actually sit down and look at the biology, which is what we're doing today, we aren't the victim. We are the fortress.
That's a great way to frame it. I mean, if you could actually see the microbial world around you, you'd be horrified.
Right.
The sheer number of microbes trying to set up shop inside you right now, billions of bacteria on your skin, spores in every single breath you take. The fact that you aren't sick right now is, statistically speaking, a miracle.
It's not luck, though. That's the point.
No, it's not luck. It's the innate host response. And that's what we're diving into today. We are taking apart the body's first line of defense. This is the system that's on watch, 24/7.
And I want to be super clear about the scope here, because I used to get this confused all the time. We are not talking about antibodies today. We aren't talking about vaccines or memory cells or B cells learning from past mistakes.
No, that's the adaptive system. That's the highly trained sniper team you call in 3 or 4 days later when things get really out of hand.
Like that.
Today we are talking about the system that is keeping you alive right now in the minutes and hours after exposure. The system that has to decide, largely on autopilot, is this friend or is this foe, without ever having seen the specific bug before.
And to do that, we've got Murray's 9th edition as our structural spine today. We are going right through Chapter 8 section by section, but we're also pulling in some great color commentary from Jowett's Chapter 8 to fill in some of the gaps.
Right. And for the medical students listening, and I know there are a lot of you, this is your bread and butter. We're going to hit the why and how that shows up on the single best answer exams. But more than that, we really want to build the mental model.
Yes. Don't just memorize the names.
Exactly. Don't memorize the list of cells. If you just memorize neutrophil equals bacteria, you'll fail the complex questions. You need to understand the logic of the war. Why does the system do what it does?
So let's map out the battlefield. We're going to do this chronologically, following the path of an invader trying to ruin your day, starting from the outside and working our way in.
Right. So phase 1 is the physical and chemical barriers, the castle walls and the moat.
If those fail,
we hit phase 2, chemical warfare. This is the stuff floating around in your blood and tissues, the soluble defenses like complement and interferons.
Which is where things get explosive.
Oh, literally. And if the chemicals don't stop them, we mobilize the troops. Phase 3 is the cellular militia. We're talking neutrophils, macrophages, and the really interesting ones, the natural killer cells.
And then finally, we'll see how this whole chaotic brawl actually sends a nice organized report up to headquarters to get the adaptive response started.
It's the handoff, the bridge.
It's a journey from the outside in. So let's stand at the gate. Section 1, barriers to infection. The wall.
The best way to win a fight is to not be in one. That is the entire philosophy of the skin and the mucosal surfaces.
Murray calls the skin the dry desert. And I think that's such a crucial reframing. We think of our skin as, well, us. Soft, maybe a little oily, pleasant to touch. But to a bacterium.
To a bacterium like E. coli or Salmonella, which evolved to thrive in the warm, wet, nutrient-dense mud of a gut, your forearm is a nightmare. It is an arid, hostile wasteland.
Okay, let's drill into why. Obviously, there's the physical brick wall, the keratin.
Right, the stratum corneum. It's essentially a layer of dead, flattened, overlapping shingles on a roof. It's tough, it's waterproof, and this is a key point. Viruses, for example, need living cells to replicate.
They're obligate intracellular parasites.
Exactly. You cannot infect a dead keratinocyte. So the top layer of your skin is a dead zone. It's a buffer. Nothing for a virus to hijack there.
But it's not just a physical wall. And this is where it gets more interesting. It's a chemical trap.
This is where the dry desert analogy gets really interesting chemically. Your sebaceous glands, the things that give you pimples when you're a teenager, are pumping out sebum. But Murray points out that this isn't just oil. It's rich in free fatty acids.
And fatty acids are, by definition, acidic.
Exactly. The pH of the skin is generally around 5.5. That is significantly more acidic than the interior of the body, which is a nice, comfortable 7.4. Most pathogens are what we call neutrophiles. They love a neutral pH.
So they land on your skin, and it's like landing in a vat of weak vinegar.
Pretty much. It inhibits their enzymes, it messes with their membrane transport. It's just a deeply unpleasant place to be.
And then on top of that, we have sweat.
Which contains lactic acid, again, dropping the pH, and salt, which can create osmotic stress. But there's another layer to the skin defense that Jowett highlights, which is the competition aspect, the normal flora.
The squatters, the microbes that are already living there.
Essentially, yes. You are covered in friendly bacteria like Staphylococcus epidermidis. It's supposed to be there. And because it's adapted to that acidic, dry environment, it occupies all the prime real estate.
So there's no room at the inn.
There's literally nowhere to park. If a pathogenic Staph aureus tries to land, the local tenants have taken all the food and all the space. They even produce their own antimicrobial peptides to kill off the invaders.
So we have a dead, acidic, salty, crowded wall. But we have chinks in the armor. We have to breathe. We have to eat. The eyes, the nose, the gut. These are the gates.
The mucosal epithelium, this is the sticky trap.
I always found this part fascinating because the strategy completely flips.
Yeah.
On the skin, the strategy is dry and dead, but in the nose and lungs, the strategy is wet and moving.
That's a perfect distinction. The body knows it can't put keratin over your lungs. Gas exchange wouldn't work. Oxygen needs moisture to diffuse across a membrane, so instead it weaponizes the wetness. It uses mucus.
Goblet cells, right?
Yep. Sprinkled throughout the epithelium, you have these goblet cells that produce mucins, these massive, heavily glycosylated proteins that just soak up water and turn it into a gel.
It's like biological flypaper.
It's exactly like flypaper. It traps bacteria, dust, pollen, fungal spores, anything you breathe in. But a trap is useless if you don't empty it. If you just catch flies and leave them there, eventually they rot or break free or pile up.
And this is the high-yield mechanic for exams, the ciliary escalator.
This is biology as engineering. It's beautiful. The epithelial cells lining your respiratory tract have cilia, these tiny hair-like projections that beat in a synchronized rhythmic wave.
They're not just flailing around randomly.
No, it's a coordinated power stroke. They are pushing that entire layer of mucus up against gravity, out of the lungs, up the trachea, towards the throat.
So we are constantly swallowing our own lung trash.
You are. Millions of times a day without even thinking about it. You swallow it and it drops into the stomach, which is the ultimate incinerator, a pH of 2. Almost nothing survives that acid bath.
I want to pause here because Murray brings up some really specific clinical correlations regarding this escalator. If the escalator breaks, you get very, very sick.
Absolutely. Think about cystic fibrosis.
That's the genetic defect in the CFTR chloride channel.
Right. And without getting too deep in the weeds, the result is that the fluid layer beneath the mucus is depleted. The mucus becomes dehydrated. It's too thick. It's like trying to pump cement instead of water.
So the cilia can't move it.
They try to beat, but they can't move the sludge. The escalator grinds to a halt. The bacteria, specifically bugs like Pseudomonas aeruginosa, get trapped. They sit there, they form biofilms, and they just destroy the lungs over time.
And what about for people without a genetic disease? What about smoking?
Smoking is a direct chemical attack on the cilia. It paralyzes them. It's a chemical stun gun. That's why smokers have that characteristic smoker's cough, especially in the morning.
Why in the morning?
Because overnight, the cilia have been stunned. The junk has pooled in their lungs. When they wake up, the only way to clear it is with a massive manual cough because the automatic system is offline.
And there's a viral connection here too, right? The idea of a secondary bacterial pneumonia.
This is a classic exam scenario. A patient gets the flu influenza virus. They feel terrible for a week. They start to get better. Then, boom, sudden high fever, cough, chest x-ray, shows a consolidation. It's a bacterial pneumonia. Why?
Because the virus paved the way.
The influenza virus specifically infects and destroys the ciliated epithelial cells. It basically rips up the carpet. So for a few weeks, until those cells can regrow, you have no escalator. The bacteria that you breathe in just walk right down into the basement of the lungs.
That is terrifyingly efficient. Okay, let's move to the chemical side of these barriers. Jowett brings up some alphabet soup here that I want to clarify. Defensins, alpha and beta.
Right. Defensins are really cool. They are basically molecular needles. They are small peptides, and they're positively charged. They're cationic.
And bacterial membranes are generally negatively charged.
Opposites attract. They attract. The defensin inserts itself into the membrane. It oligomerizes, forms a little ring with other defensins, and pop. It punches a hole, a pore, The contents leak out and the bacterium dies.
So what's the difference between alpha and beta? Jevitz makes a point to separate them.
It's mostly about geography. It's a minor point, but exams love these little differentiators. Beta defensins are primarily produced by epithelial cells. Think respiratory and urogenital epithelium.
We use a mnemonic. B for breathing. Beta defensins are in the breathing tubes.
That works perfectly. Alpha defensins are found in the granules of neutrophils, your white blood cells, and also in Paneth cells, which are these specialized cells deep in the crypts of your gut. Different locations, same basic weapon.
And then we have lysozyme, the Pac-Man of the innate system.
Found in tears, saliva, sweat, all your mucosal secretions. Lysozyme is a sniper enzyme. It targets a very specific bond in the bacterial cell wall, the glycosidic bond between NA and NAG in peptidoglycan.
This brings up a key structural difference. Lysozyme works great on gram-positives. Why is it less effective against gram-negatives?
Because gram-positives wear their heart on their sleeve. Or rather, they wear their peptidoglycan on the outside. It's a huge, thick, exposed layer. Lysozyme can walk right up and start snipping it to pieces.
But gram-negatives have that outer membrane.
Exactly. They have that lipid bilayer shield covering their thin peptidoglycan layer. Lysozyme has a harder time getting in to reach its target unless something else like complement, which we'll get to, disrupts that outer shield first.
Okay, so we've covered the skin, the mucus, the acid, and the enzymes. But let's say the bug is smart. It finds a cut in the skin, or it's tough enough to survive the stomach acid. It is now inside the tissue. The wall has been breached.
Now the game changes entirely. The bug is in the interstitial fluid. It thinks it's safe. It starts looking for food, for resources to multiply.
And this leads us to section 2, soluble defenses. And the first thing the host does is hide the silverware.
Chuckles. Nutritional immunity. I love
Sure.
this concept. It's a siege tactic. The most precious resource for a bacterium trying to multiply inside the human body isn't sugar. It's iron.
Why iron specifically?
Iron is the master cofactor. It's at the heart of the cytochromes used in the electron transport chain. It's needed for DNA replication enzymes, for almost every critical metabolic process. A bacterium cannot multiply without iron. It's a non-negotiable requirement.
So the body enacts a scorched earth policy.
Exactly. We have these proteins like lactoferrin, which is in the mucus, and transferrin, which is in the blood. These are iron-binding proteins, and they bind iron with an affinity that is almost mathematically impossible to break. They scrub the environment clean of any free iron.
So the bacterium is literally starving. But Murray mentions this is an arms race. The bacteria fight back.
They do. This is a classic host-pathogen chess match. The bacteria produce cytophores.
What are those?
These are small molecules that the bacteria launch out like grappling hooks. They are designed with an even higher affinity for iron than our transferrin. They literally rip the iron away from our proteins and bring it back to the bacterium.
It's a microscopic tug of war.
It is. And we have a countermove for that. We produce a protein called cytorocolin. Do you know what cytorocolin does?
I'm guessing it binds to the cytophore.
It binds the cytophore itself. It jams the grappling hook. It's layer upon layer of evolutionary countermeasures. This war has been going on for millions of years.
And this directly explains a clinical finding, right? The anemia of chronic disease.
Precisely. That's the clinical link. When you are chronically inflamed or infected, your liver produces a hormone called hepcidin. Hepcidin locks iron away inside your macrophages and prevents it from being absorbed from the gut. The goal is to keep it away from the bugs.
But the side effect is?
The side effect is your red blood cells starve for iron too. You become anemic. But it's actually a defense mechanism. Your body is prioritizing starving the infection over making new red blood cells.
Incredible. Okay, the bug is starving, but it's still alive. Now it has to deal with the heavy artillery, the complement system. Section 3.
This is usually where medical students start crying.
It is a nightmare of numbers. C1, C4, C2, C3b, C5a. It sounds like a list of droids from Star Wars.
Yeah.
But we're going to simplify it. We need a mental model that sticks.
The best way to think of complement is as a cascade of dominoes. You have these proteins, C1 through C9, floating around in your blood, completely inactive. They are just potential energy.
Waiting for a push.
Waiting for a trigger. And the goal of the entire system, the only thing you really need to care about initially, is knocking over the big central domino labeled C3.
The convergence point.
Yes. Murray makes a huge point of this. There are 3 ways to start the chain reaction: the classical pathway, the alternative pathway, and the lectin pathway. But all 3 roads lead to C3. Once C3 is split, the rest of the war is identical.
Okay, let's start with the alternative pathway, because Murray says this is the most ancient one. It's the tripwire.
This is fascinating because it relies on the inherent instability of the C3 molecule itself. C3 is basically a grenade with a loose pin. In your blood right now, C3 is constantly, spontaneously ticking over. A small percentage of it just hydrolyzes and breaks apart into C3A and C3B.
So our immune system is constantly misfiring. That sounds dangerous.
It's constantly idling. Think of that C3B fragment like a sticky bomb with a very short fuse. If that spontaneously generated C3B lands on one of your own human cells, we have regulatory proteins on our cell surfaces like factor H or DAF that immediately diffuse it. They say, safe, this is a friendly cell, do not explode.
But if it lands on a bacterium?
Bacteria don't have factor H. They don't have the I'm friendly signal. So the C3B sticky bomb lands and it sticks. The short fuse is stabilized by another factor called properdin or factor P. And suddenly, you have a recruitment of factor B and factor D, and you build a machine on the surface of the bacteria.
The C3 convertase.
The alternative C3 convertase, and this is the key. This machine's only job is to grab other C3 molecules from the blood and split them into C3A and C3B at an incredible rate.
So it's an amplification loop.
A massive amplification loop. One C3B that sticks by chance creates a machine that makes thousands more C3Bs, which then coat the entire bacterium in seconds. It's an explosion.
That's the alternative entrance.
Yeah.
What about the lectin pathway, the sugar sensor?
This is a bit more targeted. We have a protein floating in our blood called mannose-binding lectin, or MBL. As the name implies, it looks for the sugar mannose.
Which is a sugar found on the surface of many bacteria, fungi, and viruses, but we don't typically display it on our own cells.
Right. Our glycoproteins are usually capped with sialic acid, so mannose is a stranger signal. MBL floats around, bumps into a surface with a lot of mannose, and it clamps down. That shape change in the MBL molecule activates associated proteases, MASPs, which then start the cascade, cleaving C4 and C2.
And eventually building a C3 convertase.
Exactly. Different trigger, same machine, same goal.
And finally, the classical pathway, the targeted strike.
This is the bridge between the old guard, the innate system, and the new guard, the adaptive system. This pathway waits for an antibody.
We have the mnemonic for this. GM makes classic cars.
Perfect. IgG and IgM are the two antibody types that can activate the classical pathway. If you have an antibody that has already bound to a bacterium, the very first complement protein, C1, docks onto the Fc portion, the tail of that antibody.
So the antibody is like a laser designator for a smart bomb. C1 is the bomb.
An excellent analogy. C1 binds, its shape changes, it becomes an active enzyme, and it starts the chain by splitting C4 and C2. But again, don't get lost in the numbers. Where do we end up?
We end up building a C3 convertase.
We always end up at C3. And once we split C3, we get two critical fragments, C3A and C3B. And this is the rule of thumb for the exam. A stands for anaphylatoxin, it floats away. B stands for binding, it binds to the surface.
Okay, let's follow A first. C3A, and a bit later, C5A. They float away from the battle. Why? What are they doing?
They are the town criers. They are screaming for help. Anaphylatoxin means they cause inflammation. They act on mast cells, causing them to degranulate and release histamine. And they tell the endothelial cells of the blood vessels to dilate and become permeable or leaky.
So the redness and swelling of a bug bite?
Yeah.
A lot of that is C3A and C5A at work.
A huge part of it, yes. They're creating gaps in the blood vessel wall so that fluid and, more importantly, other immune cells can get out into the tissue where the fight is.
And C5A has a special superpower, doesn't it?
It does. It is one of the most potent chemotactic factors known. It is a scent trail for neutrophils. It diffuses out into the tissue, creating a concentration gradient, and the neutrophils smell it and start crawling towards the source, towards the battle.
Okay, so the A fragments are the alarm bells. Now let's look at B, C3B. It binds to the bacteria.
It sticks all over the surface of the bacteria, and this is vital because of a process called opsonization.
The ketchup effect.
Chuckles. Yes, we always come back to food. It's the perfect analogy. Macrophages and neutrophils are picky eaters. They have a hard time grabbing onto the slippery, often encapsulated surface of a bacterium. But they have receptors all over their surface that are specifically designed to grab C3b.
So if a bacterium is coated in C3b,
It's delicious. The phagocyte grabs it instantly and eats it. This is arguably the most important function of the complement system, opsonization. It makes the bad guys easier to eat.
but C3b can do one more thing. If enough of it builds up, it can trigger the finale, the terminal pathway, C5b through C9.
The MAC, the membrane attack complex.
This is my favorite part of the chapter. It sounds like something out of science fiction.
It is biological nanotechnology. It's incredible. The C3b helps form a C5 convertase, which splits C5a and C5b. C5b then sticks to the membrane and recruits C6, C7, and C8. They form a complex that burrows into the lipid bilayer of the bacterium.
So they're creating a foothold.
Exactly. And then C9 comes in, not one C9, but up to 16 copies of the C9 protein. They insert into that hole and they polymerize into a ring, a literal physical tube that punches right through the bacterial membrane.
It drills a hole in the bacterium.
It's a transmembrane pore. It completely destroys the cell's osmotic gradient. Water rushes in, essential ions and ATP rush out. The cell swells up and bursts. It's osmotic lysis.
But, and here's the giant high-yield warning sign for the listeners, the MAC doesn't work on everyone.
No. It is surprisingly limited. Think about gram-positive bacteria. They have that thick peptidoglycan mattress we talked about. The MAC forms on the outside of that, but it can't reach the actual cell membrane underneath to do any damage. So for the most part, it's useless against gram-positives.
So who is it for?
Gram-negatives, which have an exposed outer membrane. And specifically, one family of bacteria is the poster child for MSC susceptibility. Neisseria.
Neisseria meningitis and Neisseria gonorrhea.
They have a very thin cell wall and are uniquely vulnerable to this drilling action. And this leads to the classic board question. You have a young adult patient with recurrent episodes of meningococcal meningitis. Not just once, but 2 or 3 times. What is wrong with them?
They are missing the MAC. They have a deficiency in C5, C6, C7, C8, or C9.
Boom. You nail that question every single time. Their innate system can opsonize the bug, but it can't deliver the killing blow.
Okay, so complement is an alarm, C3A, C5A, a glue for eating, C3B, and a drill, phagocy. But with all this explosive power, how do we stop it from blowing us up?
Regulation. We have multiple redundant safety switches. We mentioned C1 inhibitor, which stops the classical pathway at the very beginning. We have factor H and factor I, which degrades C3b. And we have DAF, decay accelerating factor, on our cell surfaces, which just kicks the C3 convertase apart. These are all host proteins that shut down complement on human cells.
And if they fail?
You get disease. For example, a genetic lack of C1 inhibitor leads to hereditary angioedema. The classical complement pathway fires inappropriately, generating inflammatory peptides and causing massive, uncontrolled, and sometimes fatal swelling in the patient. It's not an infection, it's a regulation failure.
Let's shift gears. We've been talking almost exclusively about bacteria, but what about viruses? This brings us to section 4, interferons, the neighborhood watch.
Right. If complement is the star player against extracellular bacteria, interferons are the primary innate defense against viruses.
And we split them into type I and type 2. Let's start with type I.
Type I includes interferon alpha and interferon beta. These are produced by a cell that has just been infected by a virus. The cell's internal sensors, like TLR3 or RIGI, detect the presence of viral RNA or DNA.
So the cell knows it's been hijacked. It's already doomed.
Usually, yes. It knows it's going down. It knows it's about to be turned into a virus factory. But it uses its dying breath to synthesize and release interferon alpha and beta.
What's the message?
These molecules drift over to the neighboring uninfected cells and bind to their interferon receptors. The message is simple. Danger is coming. Shut it down.
What does shut it down mean in cellular terms?
It triggers an antiviral state in the neighboring cells. They activate enzymes that will degrade any viral RNA they encounter. And most importantly, they shut down their own protein synthesis machinery.
So they close the factory.
They close the factory. A virus is useless if it can't use the cell's ribosomes to make more copies of itself. So the interferons effectively create a firewall of resistant cells around the initial infection containing the spread.
It's incredibly altruistic. I'm dying, but I'm going to warn you all so you can save yourselves.
It is. It's a fundamental part of our defense. Now, type 2 interferon is totally different. That's interferon gamma.
Gamma is the general.
It is. It is produced not by infected tissue cells, but by immune cells, specifically NK cells and T cells. Its main job isn't directly antiviral. It is to scream at macrophages.
To activate them.
Yes. Arresting macrophages is like a janitor. It's ambling around, cleaning up cellular debris, but you splash some interferon gamma on it, and it turns into a warrior, an M1 macrophage. It becomes hyper-aggressive, its metabolism ramps up, and it becomes incredibly efficient at killing things it has ingested.
Which is a perfect segue. Let's talk more about those macrophages. Section 5, the cellular militia. Let's meet the troops.
First up, the neutrophils, or PMNs, polymorphonuclear leukocytes. Murray calls these the grunts of the immune system, and it's a great term.
They're the first responders, the rapid response force.
Extremely rapid. They are the most abundant white blood cell in your circulation. They are produced in the bone marrow in huge numbers, but they live fast and die young. Their lifespan is usually less than 3 days.
Now, how do they kill?
Brute force. They are essentially walking bags of granules filled with destructive enzymes. They ingest bacteria into a phagosome and then just dump everything on it. Defensins, lysozyme, and most importantly, they unleash the respiratory burst, which we will break down chemically in a moment.
And when they die, they don't just fade away, right?
No. Their death is a final act of war. They can undergo a process called mitosis. They explode, spewing out their DNA, which forms a sticky web called a net, a neutrophil extracellular trap. This net is studded with all their toxic granule proteins, and it physically ensnares and kills bacteria even after the neutrophil itself is dead.
And the accumulation of dead neutrophils in these DNA nets.
That's pus. If you see pus, you are looking at a neutrophil graveyard.
There's a clinical point here I want to hit, the left shift. What does that mean?
Right. When you have a massive bacterial infection, your body sends out the call for neutrophils. The demand is so high that the bone marrow can't produce and mature them fast enough, so it starts releasing immature form cells called band cells. On a blood smear, seeing a high number of these immature band forms is called a left shift. It's a huge red flag for an acute bacterial infection.
Okay, next up in the militia, the macrophages, the tanks.
Unlike the short-lived neutrophils, these are long-lived. They are the resident guards. They take up stations in tissues and just wait. In the liver, they are called Kupffer cells. In the brain, they're microglia. In the bone, osteoclasts.
And Murray talks about this M1 versus M2 duality. This is a really important concept.
It's the two faces of the macrophage. The cell can be polarized to do two very different jobs. M1 is the classically activated macrophage triggered by interferon gamma, as we just said. This is the killer. It produces nitric oxide, reactive oxygen, and causes inflammation to destroy bacteria.
And M2.
M2 is the alternatively activated macrophage. It's triggered by different cytokines like IL-4 or IL-13. This is the healer. It cleans up the debris from the battle, it stimulates collagen production by fibroblasts, and it helps repair the wound after the fight is over.
So you need M1 to fight the war and M2 to rebuild the city afterwards. What happens if the M1 macrophage can't kill the bug? I'm thinking of something like tuberculosis.
A great example. Mycobacterium tuberculosis has evolved to live inside macrophages. The M1s just can't clear it. So if they can't kill it, they wall it off. They change their shape, they fuse together to form giant multinucleated cells, and they recruit other immune cells to build a wall around themselves. That structure is a granuloma. It's a prison for the bacteria.
Next cell type, the dendritic cells, the scouts.
We'll touch on these more in the final section, but their main job is information gathering. They are the bridge between the innate and adaptive worlds. They sit in the tissues, sample their environment, and if they see something dangerous, they take a snapshot of the enemy and run it back to headquarters, the lymph nodes, to show the T cells what's going on.
And finally, a group that is getting a lot more attention in the 9th edition, the innate lymphoid cells, ILCs, and the NK cells.
ILCs are fascinating. Think of them as innate versions of T helper cells. They produce the same cytokines, but they don't have a specific antigen receptor. They are pre-programmed. ILC1 is like a Th1 cell. It makes interferon gamma to fight viruses. ILC2 is like Th2. It makes IL-5 and IL-13 to fight parasites. They provide a rapid early source of these critical cytokines.
So they're like a pre-packaged T-cell response.
Yeah.
And that brings us to the natural killer cells. These are the secret police.
I love NK cells. Their targeting mechanism is completely unique. They don't look for a specific criminal. They look for suspicious behavior. Specifically, they're checking everyone's ID card.
The ID card being MHC class 1.
Correct. Every healthy nucleated cell in your body constantly presents little bits of its own proteins on MHC class I molecules on its surface. It's a way of saying, I'm self, I'm healthy, everything's fine in here. The NK cell has an inhibitory receptor called a KIR that reads this ID card. If it sees MHCI, it gets an inhibitory signal, it holsters its weapon, and moves on.
But viruses are tricky. They know the T cells recognize viral proteins on MHCI, so some viruses and many tumors downregulate MHCI to try and hide.
Exactly. They try to go stealth. They pull their ID cards inside. But the NK cell sees the absence of the ID card. This is the famous missing self hypothesis. If the inhibitory signal is gone, the NK cell's activating signals take over, and it kills the target immediately using perforin and granzymes.
So T cells kill what they see, a foreign antigen on MHCI, and NK cells kill what they don't see, the absence of MHCI.
A perfect way to summarize it. It's a beautiful system of checks and balances. And they also have another trick, ADCC. Antibody-dependent cellular cytotoxicity.
That's a mouthful.
It is, but it's simple. If a target cell is coated in IgG antibodies, the NK cell has a receptor called CD16 that can grab the tail end of those antibodies. This cross-linking activates the NK cell and it kills the target. So they can also work as executioners for the adaptive immune system.
We've got the barriers, the chemicals, and the cells. But how does the system actually wake up in the first place? Section 6, activation, sensing danger.
This is the intelligence part. We mentioned earlier that the system is nonspecific, but that's not entirely true. It's broadly specific. It doesn't recognize a single strain of E. coli, but it recognizes things that all gram-negative bacteria have in common. It recognizes patterns.
And these patterns are PAMPs and DAMPs.
Right. PAMPs are pathogen-associated molecular patterns. These are structural things that bacteria, viruses, and fungi simply cannot hide. Things like lipopolysaccharide, LPS on gram-negatives, peptidyl glycan on all bacteria, flagellin from flagella, double-stranded RNA in viruses. These are conserved structures that are essential for the microbe's survival. They can't just mutate them away without dying.
And what are DAMPs?
Damage-associated molecular patterns. These are signals from your own cells when they are injured or dying in a messy way. Things like ATP leaking out into the extracellular space, uric acid crystals forming, or DNA showing up in the cytoplasm where it shouldn't be. It's the cell screaming, "I'm hurt!"
And our cells have sensors to detect both of these things.
They do. The sensors are called PRRs, or pattern recognition receptors, and the most famous family of PRRs is the toll-like receptors, the TLRs.
Okay, this is high-yield matching for the exam. We need to know which TLR sees what. Let's do the big ones.
Absolutely. The most famous one is TLR4.
TLR4 sees LPS, lipopolysaccharide.
Correct. So TLR4 is our primary sensor for gram-negative bacteria. What about TLR2?
TLR2 is more for gram-positives. It sees peptidoglycan and lipotecoic acid.
Exactly. So TLR4 and TLR2 sit on the cell surface waiting to spot bacteria. But then we have the intracellular ones, TLR3, 7, 8, and 9. Why are they inside the cell in endosomes? Because they are looking for nucleic acids from viruses. Viruses have to uncoat inside the cell to replicate. So TLR3 sits in the endosome and looks for double-stranded RNA, a hallmark of many viral life cycles. TLR7 and 8 look for single-stranded RNA, and TLR9 looks for unmethylated CpG DNA, which is common in bacteria and some DNA viruses.
So TLR4 on the surface spots the E. coli in the bloodstream, and TLR3 inside an epithelial cell spots the influenza virus that just got in.
A perfect summary. Now, there is another really important sensor inside the cytoplasm that we have to talk about, the inflammasome.
Sounds like a wrestling move.
It acts like one. It's a protein complex, the most well-known is NLRP3, that self-assembles when it detects PAMPs, or, very importantly, DAMPs, like potassium leaking out of the cell. When this complex forms, it activates an enzyme called caspase-1.
And caspase-1 is a cleaver.
It is. Its job is to find the precursor forms of two major inflammatory cytokines, pro-IL-1 beta and pro-IL-18, and chop them into their active forms. And active IL-1 beta is a master switch for inflammation.
The inflammasome can also trigger a very dramatic form of cell death called pyroptosis.
Pyro meaning fire. It's a fiery, highly inflammatory cell death. The cell literally explodes, releasing all that active IL-1 beta and other DAMPs to alert the entire neighborhood that something is very wrong. It's suicide to alert the village.
Which leads us perfectly into Section 7, chemotaxis and phagocytosis, the search and destroy mission.
So the alarm has gone off. IL-1 and TNF-alpha are being released. Now we need to get the soldiers, the neutrophils, from the highway of the blood into the battlefield of the tissue. This process is chemotaxis.
Following the scent trail.
Exactly. And the scent is made up of C5a from the complement system, bacterial proteins themselves, like F-metlufi, and a class of cytokines called chemokines, like IL-8, which is a potent neutrophil chemoattractant. The neutrophil follows this gradient.
But first, it has to get out of the blood vessel. This is diapedesis, or extravasation.
It's a beautiful three-step dance. First, rolling. The inflammatory cytokines have told the endothelial cells to put up speed bumps, proteins called selectins. These grab the neutrophil loosely, and it starts to roll along the vessel wall instead of whizzing by.
Step 2.
Adhesion. As it rolls, it gets exposed to chemokines, which activates integrins on the neutrophil's surface. These integrins are like grappling hooks, and they bind tightly to ICAMs on the vessel wall. The neutrophil comes to a dead stop.
And third, squeezing through.
Transmigration. The neutrophil literally flattens itself out and squeezes between two endothelial cells to get out into the tissue.
Okay, so now it's in the tissue. It finds the bug. Maybe it's coated in that C3b ketchup, and it eats it. Phagocytosis. Now the bug is trapped inside a bubble called a phagosome. How does the neutrophil kill it?
This is the respiratory burst, and you have to know the enzymes for your exam. It's a chain reaction.
Walk us through the enzyme chain step by step.
Step 1. An enzyme complex called NADPH oxidase. This machine sits on the phagosome membrane. It takes molecular oxygen from the cell and turns it into the superoxide radical, $2.
And this is the step that is broken in chronic granulomatous disease, CGD, right?
Precisely. If you have a genetic defect in NADPH oxidase, you can't make superoxide. Your phagocytes can eat bacteria, but they can't kill them effectively. Specifically, catalase-positive bacteria like Staph aureus or Aspergillus, which can break down the little bit of peroxide that might be present.
Okay, so we have superoxide. What's step 2?
An enzyme called superoxide dismutase takes two superoxide molecules and turns them into hydrogen peroxide, Th2O22.
And finally, the killing blow. Step 3.
The enzyme myeloperoxidase, or MPO. This enzyme is unique to neutrophils and gives them their power. It takes the hydrogen peroxide and combines it with a chloride ion to create hypochlorite.
Which is household bleach.
Literally bleach. The neutrophil bleaches the bacteria to death from the inside.
And a fun fact, myeloperoxidase contains the heme pigment, which is green. And that is why pus is green.
It is. It's the color of the enzyme from all the dead and dying neutrophils.
While this microscopic chemical warfare is happening, the battlefield itself is heating up. Section 8. Inflammation. The fire.
Inflammation is the necessary collateral damage of the war. It's driven by the acute phase cytokine triad, IL-1, IL-6, and TNF-alpha. These are released mainly by macrophages that have detected PMPs.
Let's assume our listener is an exam candidate. What does each one do that they need to remember?
IL-1 and TNF-alpha are the major pro-inflammatory signals. Locally, they make blood vessels leaky and sticky, as we discussed. Systemically, they act on the hypothalamus in the brain to cause fever. They are endogenous pyrogens.
And what about IL-6?
IL-6 is the main messenger to the liver. It tells the liver to stop what it's doing and switch to wartime production. It needs to pump out acute phase reactants.
Like C-reactive protein, CRP.
CRP is the classic one. But also fibrinogen, which can cause red blood cells to stack up and increases the ESR. And hepcidin, which we mentioned earlier, to hide the iron. If you measure a high CRP in a patient, you are measuring the liver's response to IL-6.
But inflammation can go wrong. It can be too much of a good thing. Murray talks about sepsis and the cytokine storm.
This is when the local response goes systemic. Imagine you have a gram-negative infection that gets into the blood. That means LPS is circulating everywhere.
So macrophages in your spleen, your liver, everywhere are hitting their TLR4 panic button at the same time.
And they all start pumping out TNF-alpha. And TNF-alpha causes systemic vasodilation. If that happens in your finger that has a splinter, it gets red and warm. If it happens in every blood vessel in your body at once, your blood pressure completely bottoms out.
Hypotension.
Shock. That's septic shock. Your blood pressure plummets, you can't perfuse your vital organs, and they start to fail. TNF-alpha is the primary villain in septic shock.
And C5a from the complement system helps it along.
It does. It adds to the vasodilation. It is a catastrophic system-wide failure. It's the fortress's own defense systems burning the castle down.
Okay, assuming we survive the acute battle, we need to make sure we remember this enemy for next time. This brings us to the finale, Section 9, the bridge to adaptive immunity.
This brings us back full circle to the dendritic cell. We called it the scout. Murray uses a great metaphor here. The clam.
The clam.
Think of an immature DC sitting out in the tissue. It is like a clam with its shell open. It is constantly filter feeding. It's drinking in the surrounding fluid, sampling everything, self, proteins, debris, and hopefully any pathogens. This is called macropinocytosis.
Just tasting the water.
But the moment its TLRs detect a PMP, say LPS, the clam snaps shut.
Activation.
It's a maturation signal. It stops eating, it pulls up its anchor, and it starts migrating through the lymphatic vessels to the nearest lymph node.
And what happens on that journey?
While it travels, it processes the antigens it ate. It chops up the bacterial proteins into little peptides and loads them onto MHC molecules. It also upregulates costimulatory molecules on its surface, like B7.
When it gets to the lymph node.
It presents that specific antigen on its MHC to a naive T cell that happens to have the right receptor. But here is the crucial part. The DC also releases cytokines that tell the T cell what kind of enemy it is.
It provides context.
Exactly. The innate system doesn't just wake up the adaptive system, it instructs it. If the DC sensed a virus via its internal TLRs, it will release IL-12, which tells the T cell, become a Th1 fighter. We need you to help kill infected cells. If it sensed a big parasite, it might release IL-4, saying, become a Th2 fighter. We need antibodies and eosinophils.
That is the big takeaway. The innate system isn't just a brute force wall. It's the strategist that sets the rules of engagement for the rest of the war.
It buys time, it contains the threat, and it gathers and delivers the critical intelligence needed for the targeted, specific, and final victory by the adaptive system.
So we have built the fortress, fought the battle, and sent the report to headquarters. Before we get to our learning reinforcement, I want to leave you with one final thought. Murray touches on this idea of the arms race.
Right. The Red Queen hypothesis from Alice in Wonderland. You have to run as fast as you can just to stay in place. Our innate system is ancient. TLRs are found in fruit flies, but bacteria are evolving every 20 minutes.
They're learning to hide their PMPs. They're making capsules to hide from complement. They're developing enzymes to neutralize our respiratory burst.
And what's really fascinating is that we are now learning that our innate system might be more trainable than we thought. There's this emerging field of trained immunity, the idea that an earlier infection can make your macrophages, these supposedly non-specific cells, better at responding to a different infection later on.
So they have some kind of memory.
A kind of epigenetic metabolic memory. It's not the specific memory of a T cell, but it challenges the dogma that the innate system is static. But that is a deep dive for another day.
For now, let's make sure you remember the essentials from today's chapter. It is time for the learning reinforcement. I'm going to ask a question. We'll pause for a second so you can think of the answer, and then we'll reveal it and explain why. These are all SBA-style single best answer.
Ready. Let's go.
Question 1. A 19-year-old college student presents with his third episode of Neisseria meningititis infection. A workup for an immunodeficiency is initiated, a defect in which component of the immune system is most likely responsible for his recurrent infections.
Think about the unique vulnerabilities of this specific bug.
Answer. Components of the membrane attack complex, so anything from C5 to C9.
Why? Neisseria is a thin-walled, gram-negative bacterium, making it uniquely susceptible to lysis by the MAC. While other complement functions are important, the MAC provides a critical killing mechanism against this specific pathogen. Patients with C5-C9 deficiencies have a dramatically increased risk of recurrent Neisserial infections. You can review this in Murray Chapter 8 in the section on the terminal complement pathway.
Question 2. A neutrophil ingests a Staphylococcus aureus bacterium. To kill the microbe, it initiates the respiratory burst. Which enzyme is responsible for the very first step converting molecular oxygen into the superoxide radical?
This is the key initiator of the whole process.
Answer. NADPH oxidase.
Why? This is the rate-limiting and initial enzyme in the oxygen-dependent killing pathway. A defect in this enzyme complex is the cause of chronic granulomatous disease, CGD, where phagocytes can ingest but cannot effectively kill catalase-positive organisms. This is covered in Murray Chapter 8 under phagocytic responses and oxygen-dependent killing.
Question 3. A macrophage in the liver encounters fragments of a gram-negative bacterium. Which toll-like receptor, TLR, is primarily responsible for recognizing a lipopolysaccharide, LPS, on the outer membrane of this bacterium, triggering an inflammatory response?
Think about the location of the PMP and the location of the receptor.
Answer. TLR4.
Why? TLR4, in conjunction with its co-receptors like MD2 and CD14, is the specific pattern recognition receptor for LPS, the major component of the gram-negative outer membrane. Remember, TLR2 recognizes peptidoglycan from gram-positives, and TLRs 3, 7, 8, and 9 are internal and recognize nucleic acids. Check out Table 8.3 in Murray Chapter 8 for a great summary of this.
Question 4. A patient is in the ICU with septic shock. Which pro-inflammatory cytokine, produced largely by activated macrophages, is considered the primary mediator of the systemic vasodilation, hypotension, and vascular permeability seen in this condition?
Which cytokine is the main villain in septic shock?
Answer. TNF-alpha, tumor necrosis factor alpha.
Why? While IL-1 also causes fever and inflammation, TNF-alpha is the potent driver of the hemodynamic collapse and multi-organ failure that characterizes septic shock. This is why anti-TNF therapies were initially trialed for sepsis, though with complex results. This is discussed in Murray Chapter 8 in the section on pro-inflammatory cytokines.
Question 5. A cell becomes infected with a virus that, as an immune evasion strategy, downregulates the expression of MHC class I molecules on its surface. Which innate immune cell type specializes in recognizing and killing these missing self cells?
They're not looking for something foreign, they're looking for something that's missing.
Answer: natural killer, NK cells.
Why? NK cells operate on a balance of activating and inhibitory signals. The binding of their inhibitory receptors to MHC class I is the dominant don't kill signal. When a cell loses MHCI, this inhibition is lifted, the activating signals win, and the NK cell kills the target via perforin and granzymes. You can find this in Murray Chapter 8 in the section on natural killer cells.
Question 6. During an acute bacterial infection, the liver is stimulated by IL-6 to produce acute phase reactants. One of these is C-reactive protein, CRP. What is the primary function of CRP in the innate immune response?
It's not just a passive marker of inflammation, it's an active participant.
Answer: it acts as an opsonin and can activate the classical complement pathway.
Why? CRP binds to phosphocholine on the surface of many bacteria and fungi. This coating, or opsonization, enhances phagocytosis. Furthermore, C1q of the complement system can bind to CRP, activating the classical pathway even in the absence of antibody. This is detailed in Murray Chapter 8 under the acute phase response.
Question 7, last one. As part of the strategy called nutritional immunity, the host produces several proteins that limit microbial growth by sequestering iron. Which soluble innate defense molecule found in high concentrations in milk and mucosal secretions performs this function?
It's a war for resources.
Answer: lactoferrin.
Why? Lactoferrin has an incredibly high affinity for iron, binding it up and making it unavailable for bacteria that desperately need it for their metabolic enzymes. This is a key soluble defense mechanism in mucosal surfaces. You can find this in Murray Chapter 8 in the section on soluble components.
If you missed any of those, don't worry. Just go back to Murray Chapter 8. The section headers map directly to these questions. It's a dense chapter, but it's the foundation for everything that comes next.
The innate system is your constant guardian. It never sleeps, it never forgets its programming, and it is the only reason you can walk through a world of microbes and stay alive.
Thanks for listening to The Deep Dive. Keep those barriers intact, and we'll see you in the next episode.
Stay healthy.